Good day, dear students. I hope today's lecture will be much more clear in terms of sound, but still some noises will happen. So today will be quite a long lecture because we finally will be talking about how to assess some cytotoxicity. Not even after incubation of your cells with nanoparticles, but just some general approaches to assessing cytotoxicity. There will be a lot of methods and so on, but first we need to discuss the results of your previous test. And obviously it was one of the most complicated because it's associated with a lot of biomedicine things, and since most of you are chemists, partially biologists, it was not as easy, and that's why the results are not as good as I want. But it was predictable. So let me just open like this. So again, if you have any questions, you can put them in chat. Okay, the first question was about ways for nanoparticles to penetrate epithelial layers. So it's definitely transcellular transport or paracellular transport because well, through cells or through tight junctions. There is no such tight junctions closure or basement permeability. It's just again random set of words. So two correct answers here. I showed them in lecture. What information about toxicity can it give you cell cultures? So it's some important information already in question because it's about toxicity, not cytotoxicity. So you need to distinguish cytotoxicity from just toxicity. And we will be talking about this today. Yeah, finally. It was 1 hour of complete silence, and just as I started lecture, they started some. So, what information we cannot obtain using cell cultures? We cannot obtain some thrombosis chains because thrombosis is a complex reaction of organism, and we cannot obtain some thrombosis chains using just cell cultures. Even if we incubate our nanoparticles with platelets, which in some ways are responsible for formation of thrombosis in organism, it cannot give us actual thrombosis chains in organism. And obviously we cannot observe some biodistribution because biodistribution is a feature of a whole organism, not of some separate cell cultures. But all of other things we can observe using cell cultures. Hemolysis, it's a degradation disruption of red blood cells, so we can just incubate red blood cells with nanoparticles under physiological condition and observe some changes. We can definitely observe some genotoxicity, and in some ways we can observe even immune response because immune response on cell level can be expressed as release of cytokines or chemokines. So all of this can, rest of these things can be observed on cell level. The next one, again, a bit tricky question. Inhaling of nanoparticles can lead to the accumulation in which organs? So it definitely lungs. And what's next? Well, it was an option without knowledge of the exact physical and chemical characteristics. It's impossible to predict where they will accumulate. Yes, it's true. It definitely we cannot predict, but at least we can suggest, because if they entered lungs, they will already be covered with some proteins, with mucosa, with some other things. And probably this will help to eliminate them with liver or spleen. You also, next question about kidneys. So kidneys, in most of the cases, not the option. Nowhere it definitely will accumulate, at least in lungs, and everywhere, well, it a bit a bit bold to say everywhere, because we have so many organs and tissues in our organism, and for example skin. There's no much nanoparticles will accumulate in skin after inhalation, definitely, and we have some other tissues which are not accumulating nanoparticles at all. So definitely not everywhere, not nowhere, but lungs, liver, spleen, and well, there's also some options that we cannot be sure. About kidneys, almost no one give a right answer to this question, because probably you do not understand the question. Again, the question was, why nanoparticles with a size comparable to 5 nanometers go predominantly to the kidneys? The question was not why kidneys filtrate such particles. The question why they go to kidneys. So in other words, why they can bypass all of other organs, liver, spleen, maybe lungs, and they will accumulate in kidneys. The right answer was because liver and spleen cannot filter nanoparticles with so small size. They filter nanoparticles with relatively large size. Because of this, nanoparticles with size about 5 nanometers will go to kidneys. There was no question about why they will filtrated by kidneys. So maybe just one person answered correctly, the rest of you answered not correctly. So please read questions very carefully. Next one, to my surprise, almost all of you succeeded. So this is a liver sinusoid, and well, there was some options, and as you can see, there's 3 non-option, but it was enough to pick just one non-option. So for example, for epithelial cells. So number 1, it's a hepatocytes, a layer of hepatocytes. 2, it's a sinusoid itself. There's some leukocytes in liver. I didn't mention this in lecture, but picking a right leukocyte number 5 will give you 2 points instead of 1. It's endothelial cells, because this is a vessel. All vessel walls covered with a monolayer of endothelial cells, not epithelial, endothelial cells. So number 3, endothelial cells, and the last cell type here is Kupffer cells, number 4. So almost all of you succeeded with this question. Well, part of you give a right answer, part of you not. So it was quite easy if you ask me, because you can just Google the answer. What is the macroscience of inflammation and why they occur? So microscience of inflammation I mentioned in lecture. It just pain, heat, redness, swelling, and loss of functions. 3 of this happens because of phosphorylation, so enlargement of blood vessels, and the rest of this connected with, well, loss of function connected with inhibition of muscles partially, and the pain also connected with inflammation. So most of you in some way gives a correct answer, but still some of you not. How can nanoparticles penetrate the spleen? So I talked a bit about spleen and filtration of different cells, for example red blood cells by spleen. And as you remember, there's a very tight space between epithelial cells or endothelial cells in spleen, and to enter this very tight space, cells should mimic shape and size, definitely. And since there's a lot of macrophages in spleen, they definitely need to mimic some chemical receptors to eliminate uptake by macrophages. And to be honest, there was 5 options, and only 3 options makes some sense, because blood cells granulation, well, it definitely some granulation of cells, but there is no way to mimic this granulation, and there is no zeta potential of cells at all. So it was just 3 correct options out of 5. Which part of the spleen is filled with blood? It's red pulp, so I mentioned it in the lecture also. And nanoparticle lead to cell death leads to inflammation ends with recovery and fibrosis. And what is fibrosis? So every option is correct. It's all of this is some kind of synonyms. So just every option. And the last one about lungs. And again, some of you probably do not understand what I talked to you during the lecture, why a lung, being not a filtering organ, so not a part of mononuclear phagocytic system, still they can accumulate nanoparticles. So the answer is because of large blood exchange, a lot of capillaries, and a lot of interactions between blood and nanoparticles in blood and different kind of cells. They can accumulate nanoparticles. So some of you answered correctly. So if you don't have any questions regarding your test, we can proceed with the lecture. And do not forget, today we also will have a test somewhere in the middle. Okay, no questions. No questions. So we can proceed. Well, today we'll be talking about different ways to assess cytotoxicity, in other words, viability of cells or cell death. Because of this, I need to give you some information about the concept of cytotoxicity. So basically, cytotoxicity, it's toxicity to cells. And you need to distinguish cytotoxicity and just toxicity, because toxicity in general can occur on the scale of a whole organism. So it just can be a general toxicity. So cytotoxicity connected with 2 types of action. And the section connected with drugs, cytotoxic drugs and cytostatic drugs. Currently, there's not much talks about cytostatic drugs, because it cannot be called drugs after all. For example, nitric oxide can be called cytostatic compound because it can inhibit cell growth. So cytostatic is something that causes stasis. So it can freeze cells and prevent them from proliferation. And this state of stasis some way can lead to apoptosis. And different from this one, it's cytotoxic drugs. And cytotoxic drugs induce a process of necrosis. But so necrosis is something bad, yeah, when the cell is dying in some uncontrolled manner, and apoptosis is some process of cell death when cell dying in some controlled manner. But in general, if you will read a very good paper, for example in Cell, in Nature Nanomedicine, in some other journals from very good publishers, you will probably see some other terms other than apoptosis or necrosis. Because what's so special about, like maybe 20, maybe even 30 years ago, it was like apoptosis is a regulated process of cell death, and it was necrosis like unregulated process of cell death. So some kind of accidental cell death. But now, if we look even on recommendation from 2015, it's cell death subtypes, and it can be regulated cell death and accidental cell death. And both apoptosis and necrosis goes to regulated cell death. What is accidental cell death? Well, accidental cell death is when you, for example, spill some acid on your blood and just dissolve some part of your epithelial cells. It's an accidental cell death. So basically, it's a damage by physical stimuli or by some chemicals and so on. All of other cell death types, it's a regulated cell death. And there's some subtype in regulated cell death, so it's RCD. And it can be also programmed cell death, so PCD. And for example, apoptosis, it's a PCD type. But just look on this recommendation. It's a review, but it's a recommendation, and just look on amount of affiliation and amount of authors. It's like 3 pages of authors and affiliation in this paper, but it's relatively old in terms of science. And so, well, I need to discuss one important thing here from the same paper about how we can distinguish when we can talk about regulated cell death or no, we cannot talk. So we can or we cannot. Previously, everything was connected with different morphological changes, damage to membrane and so on, but for now a lot of things connected with the level of ATP and reactive oxygen species. So ATP is a main molecule which carry energy in our cells, and this level of ATP correlated with probability of cell survival. So, for example, we have some homeostatic perturbation. What can be homeostatic perturbation? It can be some drugs, it can be some nanoparticles, since we are talking about nanotoxicology. And during this perturbation, cell can enter adaptive response phase. During this adaptive response, for example, nanoparticles can trigger some release of reactive oxygen species, but and decrease of some ATP, and vice versa. But after some point of time, cell have some limits, so it cannot resist this damage for eternity, and after some time ROS amount will increase and ATP will decrease. At the specific point called point of no return, it will be execution of regulated cell death. So there is no way for cell to survive if ATP level pass this border. So this is one of the most important criteria to regulated cell death. And if we're talking further to a recommendation from 2018, it's a lot of different types of regulated cell death. So if you look here, it can be necroptosis, pharoptosis, we talk about this, peroptosis, paranatosis, entotic cell death, netosis, netotic cell death, and a lot of other type of cell death. And for example, extrinsic apoptosis, intrinsic apoptosis, and so on. And when we observe, for example, our cells after incubation with some simple nanoparticles, there is no way with using just a simple technique like fluorescent staining understand what really happened to these cells after incubation with nanoparticles. And to understand what is the real cause, what is the real type of regulated cell death we observe after incubation with our nanoparticles, we need to perform some polymerase chain reaction, so real-time PCR, to understand which genes are upregulated, which are downregulated, because there's a lot of patterns which can be observed during these types of cell death. And as you can see, regulated cell death can possess both necrotic morphology or apoptotic morphology, and in some of these types of cell death, we can observe both of them. So to be correct, if you aim to publish in some good journals, you should avoid such terms as apoptosis or necrosis, because you need to prove that you really observe such kind of cell death. Probably you should, if you just stain them with some fluorescent dyes, you should just use some cell death, well, or some terms as viability or cell survival, without mention a type of cell death. And again, this regulation from 2018, and as you can see, there is a lot of authors. It's like 200 affiliations, and it's just interesting that both of this recommendation and this recommendation, first author is Lorenzo Galuzzi, and for you just to wonder a bit, it's most cited and most like you see H index 117. It's most cited and one of the most important scientists, biologists in Europe. And I believe he likes 40 years old. So, well, everything connected with cell death and cell death pathways is very important in biology. But let's now jump to cell types and what can happen to cells and how to understand what damage nanoparticles can cause to your cells. So it's a lot of different type of cells, like it can be HeLa cells or HepG2 cells. It can be some epithelium cells of different animals. It can be cancer cell lines from humans. It can be even cells from insects, like some worms. It can be cancers of animals like mouse glioma. It can be human neuroblastoma cells and so on. It can be unicellular algas. It can be plant cells. And among all of these cell types, there are some similar processes which are connected with, let's say, response to nanoparticles introduction. So some similar process and some processes are not very similar. And because of this, we need to understand how to make some images, visualization of this process, or how to understand if this process occurs or not. So, what can be the target of nanoparticles? We need to understand this because if, for example, you synthesized some nanoparticles and you add these nanoparticles to your cells, and now you need to stain your cells to understand what's bad happened to your nanoparticles, or to your cells. But before this, you need to predict, to suggest what are the possible target of your nanoparticles inside cells. And there's a lot of targets. So nanoparticles are one of the first type of cytotoxicity. It's a formation, generation of reactive oxygen species. Some damage can be done to endosomes or endolysosomes. Some damage can be done to DNA. Some damage can be done to membranes. Some damage can be done to mitochondria, and a lot of things. Some nanoparticles can bind to receptors on the surface of the cells, and a lot of things. And in most cases, it's very hard to predict what will happen to your nanoparticles when they enter your cells, and even harder to predict what can happen if they enter a whole organism. So I will show you just a bit of example, a very nice video which were recorded a lot years ago about gold nanoparticles inside a living macrophage. So as you know, macrophages can uptake, like Kupffer cells, they can uptake nanoparticles from bloodstream. And how this looks? So it looks something like this. So it's a macrophage, one macrophage, second macrophage, it's a dead cells. And this is a gold nanoparticles inside the macrophage. So another macrophage here. And they're just eating dead cells because it's their duty. Maybe it's not a macrophage. I believe maybe it's neutrophil of some kind. But anyway, it's some type of immune cells. And all of these gold yellowish dots, it's gold nanoparticles, aggregates of gold nanoparticles, which were visualized using a dark field technique on optical microscope. So as you can see, it's a macrophage, let's call it macrophage. Macrophage with some gold nanoparticles inside. But where's inside? Well, no one knows. So let's go to some ways to assess cell viability, and we will start from direct ways to assess cell viability. You already got this question in your introductory test, and not of you understand what is direct way and what is indirect way. So just for you to understand, direct way is when you observe some damage or some cytotoxicity with your own eyes, and you can definitely say what is the cause of this toxicity, and you can observe the toxicity outcomes. So I will explain. For example, the first one is morphology. So morphology, basically, it's a shape and structure of the cells. And cell morphology strongly depends on viability. For example, this is 3 pictures of the same cell culture at the different stages of survival. So 100% viability, perfectly healthy cell culture, 70% of viability, and 40% of viability. What are the main differences here? It's adherent cell culture, so all of the cells should be adhered to the surface of Petri dish or culture flask. And most of the cells are adherent here. But if you look on 70%, not most, but a lot of cells are spherical in shape, and this means they detached from the surface. And since they detached from the surface, I mentioned this in previous lecture, they lost some connection to the surface, and this is not very beneficial. This is very bad for cells. So they lost this connection, but only observation on these morphological changes cannot give you an answer why this happens. So why these cells lost this connection? Because of damage done to, for example, cytoskeleton, because of depletion of ATP, because of direct damage to these collagen fibers which hold the cells in place. You cannot answer this question. You can just say that something bad happening to cells because some of them are detached from the surface. And 70%, it's not a big decrease in cytotoxicity, but still it's a lot of cell detached, and probably they will die. So you can wait for a huge decrease, bigger decrease in if you will incubate your cells for a longer period of time. So it happens after some time you will observe only a 40% viability, so it's a 60% decrease in viability, and there's almost no cells visible, but still we obtain some viability percent. Why this happens? Because this percent obtained not from direct observation. This percent obtained from common MTT assay, and we will talk about MTT assay a bit later. But just compare images. So 70% cells are living, but if you look on this picture, it's not 70% of cell adherent. It's like maybe here some adherent cells, here some adherent cells. So according to this image, much more than 70% of cells are dying. And on this image, you even cannot observe some cells, but according to MTT, 40% of cells survived. So it's some kind of misleading between these two observations. So here like 100% of cells dead, and according to MTT, still 40% survived. And we will discuss it in details. For what purposes you can use direct observation with microscope? To count total amount of cells, definitely. To observe some morphological changes like size and form, it's still good to observe some changes, because this observation sometime can be handy. And you also can count amount of adherent cells. In some ways it also can be useful, but that's all basically that you can obtain from a simple optical microscope. But there's some other more powerful techniques which are also connected with the shape and size of cells, not only but also connected with shape and size. It's a flow cytometry, and flow cytometry, I wouldn't talk in details about flow cytometer, but still you can, for example, here you need to count with your own eyes or with some special programs, but still you need to obtain some images, and using flow cytometer you can count thousands of cells. For example, like this, I believe some maybe 30,000 or 40,000 of cells can be counted. And what is so special about flow cytometry? All of the cells, just look on the cells, they have some internal structure. So they have some organelles, nucleus, some granulation, and this granulation depends on morphology and also depends on, for example, stage of cell death. So on some stages of cell death, granulation will increase because of formation of apoptotic bodies. And this granulation can affect two parameters of your cells. It's a side scattering and front scattering. So flow cytometer, as it's mentioned in the name, flow, you have a very thin capillars. In these capillars, your cells will flow one by one, and with a special laser, you will illuminate your cells and read two parameters, side scattering and front scattering. And based on these parameters, you can understand granulation, size, and other parameters of your cells, and put your cells in different subsections, subpopulations on this plot. And even without any fluorescent staining, you will understand, for example, these cells in apoptosis, these cells in necrosis, and this is not cells after all, this is just already part of cells. So flow cytometry is a very powerful method to understand a stage, different subpopulations in your culture, cell culture. But basically it's not a flow cytometry, it's fluorescent. In most cases you need to use some fluorescent dyes to use a flow cytometry. But so let's go to fluorescence. So fluorescence is another type of direct way to assess cell viability, because for now, with using of fluorescent drugs, we can stain almost everything. And just a couple of words about basic principles of fluorescence. I believe all of you know, but still, if we have some molecule, this molecule have some energy levels, and among these energy levels, there can be different energy states. And if we illuminate this molecule with some light, with a specific wavelength, we can charge this molecule, and when discharge happens, it can be non-radiative transition and radiative transition called fluorescence. So not all of the molecules have fluorescent properties, because some molecules have only non-radiative transition in their structure, but still some molecules have, and these molecules called fluorescent dyes. Some of them called fluorescent dyes. In most of the cases, when you have some absorption peaks, so it's a feature of some fluorescent molecule. Fluorescent molecules have some absorption peak and emission peak. What does this mean? For example, to provide some fluorescence, you need to illuminate your molecule with a specific wavelength. And after this illumination, a part of energy will be lost because of some dissipation, some heating, and so on. So, since some energy will be lost, some of the energy, the remaining energy, which will go to fluorescent, will have, well, the energy is lower, the wavelength is bigger. So it's a dependence, I don't have a formula here, but you just need to remember that the higher the energy, the lower the wavelength. The lower the energy, the higher the wavelength. And this is called a Stokes shift. So if you eliminate some of your molecules with blue, you will observe probably a green fluorescence or red fluorescence, not vice versa. But still, sometimes we can observe opposite anti-Stokes shift, and this is one of the example of this anti-Stokes shift, which can be observed using up-conventional nanoparticles. So up-conventional nanoparticles usually incorporate some rare earth elements like cerubium or europium, and how these nanoparticles works. For example, you illuminate these nanoparticles with infrared light, so red light, and you can observe some visible light or even ultraviolet light in some ways. And these nanoparticles can be very useful, for example, to image something in organism, because our organism and our skin are not very good penetratable for visible or ultraviolet light. So, it's just the basic principles. But when we are talking about staining of your cells, so when we are coming to some experiments, we need to answer a lot of questions. And usually when you are just a chemist and you just synthesize some nanoparticles and you come to cell biologist or just biologist and ask, well, I have some nanoparticles, I need to add them to cells and I need staining, some fluorescent staining to see what happens. And well, it's a very complicated question because it's a lot of sub-questions need to be answered to understand what you want to see. And there is a lot of ways to stain cells. It's enormous ways to stain cells. And the main question is, first one of the most important, what do we want to stain? It's a lot of different options to stain. You can stain, for example, different centrioles, like shown here, different chromosomes or cytoskeleton. You can combine this image to observe a whole picture of cell division, for example, and it's some kind of process. It's a cell division. It's a mitosis. And the second question is: do we want to see this process, or we want to see some, I don't know, just chromosomes, for example? What kind of sample quality do we need? So is this just a routine investigation, or we want a top high quality for Nature journal? Will we observe the sample again? Do we need to save the sample? Do we need to fix this sample or store this sample for a longer period of time? What kind of fixation should we apply? Because some kind of fixating agents can interact with your nanoparticles. And there's a lot of technical questions. What is the concentration of the dye? So this is a question which are not which should be answered by cell biologists, not by chemists with nanoparticles, but still it's a lot of different technical questions. So what is the concentration of the dye, what is the time of staining, how, and one of the most important again, how to interpret the result, because sometimes results can be quite, well, quite complex. And the next video, I believe, it's some example of cell process. So when we are talking about process, usually when you stain some cells, it's a fixed cells. So you fix them with some paraformaldehyde, for example, and or other type of fixating agents, for example, methanol, and there is no way to observe some process. But sometimes it is important and it is necessary to observe some process, and current state of technology allows us to stain, for example, phagocytosis, pinocytosis, and so on. So this example of phagocytosis, sadly without staining, but still some impressive example. So, and we can observe this with our microscopes. I believe I need to switch it off, like division and home. So as you can see, it was a single cell. Now it goes to mitosis. It's a division, telophase, and there's some kind of particles, and just compare the size of the cell and the size of particle. So phagocytosis, home. Just for you to understand how big can be nanoparticles compared to cells, and still the cells can phagocyte them. So phagocytosis is really for really big objects. And let's talk a bit in details about fluorescent staining. So fluorescent staining, usually called fluorescent differential staining, because it used to differentiate some cells from another cells, or dead cells from living cells, or I don't know, some subpopulations from another subpopulations. So because it used to differentiate something, it called differential staining. And one of the most common type of this differential staining is so-called live-dead staining. So differentiate live cells from dead cells. One of the most frequently used is annexine 5 feeds plus prapidium iodide. First one here. And what's so important, as you can see, prapidium iodide used everywhere because it's the most common staining for dead cells. So how it's working? Very simple. Prapidium iodide can enter only damaged cell membrane. So there is almost no way to prapidium iodide to stain living cells. And after prapidium iodide enter damaged cell, it can bind to DNA and produce some red fluorescence, red or yellowish fluorescence. So what is annexine? What with annexine labeled feeds? So feeds produce some green fluorescence, but when we conjugate feeds with annexine, annexine can specifically binds to phosphatidylserine, and this phosphatidylserine usually on the inner surface of cell membrane. But when cell enter in apoptosis or other type of cell death, not even cell death, let's say if some damage occurs to the cell, this phosphatidylserine switch from inner side of cell membrane to outer side of cell membrane. And so macrophage or other immune cells will know that something bad happened to the cell, and they will phagocyte this cell. But in laboratories, these annexine labeled feeds, which can bind to phosphatidylserine, used to understand is this cells undergoes apoptosis or not. So using just one set of dyes, you can distinguish three types of cell death. So, for example, here you see cells with a red core and with some green fluorescence. What this means? So, cells at this stage, for example, red core, it means that they have damaged membrane. And green fluorescence mean that they still have some apoptosis going. So they are in the middle of dying. In case we see only a red fluorescence, it means it's a late state of apoptosis and phosphatidylserine on the surface of membrane already destroyed or something happened to it. And in case we see only a green fluorescence without a red nucleus, it means that cells on the very early stage of apoptosis. So just using one kit, we can distinguish three stages of cell death. So early, some middle, and late stage. Other kind of staining, for example, acadine orange. Acadine orange can be used to stain both DNA and RNA because it's this staining have a feature of multicolor staining. So acadine orange stain DNA in green and RNA in red or orange fluorescence. So in case we have some damage done to cells, almost all of the cells start dying from destruction of DNA. So, well, it's destruction done to DNA and nucleus. It's almost for every cell, it's a first stage of cell death. And because of this, if we stain our cells with acadine orange and the cell nucleus stained with red, it means that some DNA in nucleus in a single chain form. And as you can know, in nucleus DNA should be in double chain form. So something bad happened to DNA, probably this cell will undergo some apoptosis, for example. So acadine orange can be used for this purpose. And also one of the most used example of a couple of dyes which used for flow cytometry also, it's a FDA, fluorescent diacetate, and prapidium iodide again. Fluorescent diacetate can stain living cells in green. Again, prapidium iodide can stain cells nucleus in red, and they are frequently used for flow cytometry for the last, 50 years maybe. So it's some kind of very simple staining just to understand is your cells dead or not, but they give no answer why they died. So to understand why they dying, you need some very specific staining. And since we talked a lot about reactive oxygen species, there should be some ways to stain them. But as you understand, there is no way to stain some ions because these species, they are ions, some radicals. There is no way to stain some, just a couple of atoms, yes. But still, you can use their ability to oxidize molecules, other molecules, to visualize some outcomes of production of reactive oxygen species. So what I mean, one of the most common dye for observing ROS accumulation is DCFDA or sometimes dehydro-DCFDA. How it's working? So do not mess just FDA with DCFDA because they work in completely different ways. Let's look. Control cells, it's just a bright field stained with prapidium iodide. Almost nothing here because all of the cells are, well, living. And because of this, on FDA staining, we see a lot of green cells. But if we add some hydrogen peroxide is highly toxic as you know, and so we see a lot of dead cells, almost no living cells. But if we add some inhibitor of hydrogen peroxide, again we will see no dead cells, a lot of living cells. But on the opposite side, how working the CFDA? We have DAPI staining instead of prapidium iodide, but still on control cells we see almost no staining with the CFDA because there's not so much reactive oxygen species inside the cells. But when we add hydrogen peroxide, we see a lot of staining. And again, opposite results to what we see at FDA group, no staining when we have some inhibition. So how this DCFDA works? Very simple. We have some dehydro-DCFDA which enter the cell membrane, and after some cellular esterases cut part of this membrane, it's still non-fluorescent, but if we have some, for example, reactive oxygen species, and also if we have, for example, some ferrocyanins, this reactive oxygen species can cut these two hydrogen atoms, like this one, and as you can see, like this one, and this molecule become fluorescent. So for some example from our lab, it's magnetite nanocontainers. As you can see here, all of these black dots is magnetite nanocontainers, and this is the CFDA staining of control cells. Well, a couple of cells are probably dying, because when cell dying, there is no way for the cell to preserve a low concentration of reactive oxygen species. So it's well, when they say dying, everything is broken, and system for eliminating of reactive oxygen species broken too. So dying cells usually have a lot of ROS inside. But after incubation with a very high amount, as you can see here, it's a lot of nanocontainers here. After incubation with these nanocontainers, we still see a lot, not a lot, just a couple of like dead cells, let's say, cells with reactive oxygen species. And this is positive controls with a low amount of hydrogen peroxide and a high amount of hydrogen peroxide, just to be sure that we perform this test correctly. So that's how it's working. But let's talk about other manifestation of ROS. It's a lipid peroxidation. So it's a body pie staining, and body pie, it's a whole family of different molecules which can be used to stain a lot of different organelles, part of the cells. And body pie with this particular wavelengths of excitation and emission, and this particular type of body pie can be used to stain peroxidized lipids. Very useful to understand, do you have some damage done to the lipids of lipids in membrane of your cells. And how this looks? Well, very easy looks. Untreated cells, it's a cells treated with cold atmospheric plasma, like here cold atmospheric plasma, which induce membrane oxidation to treat some glioblastoma, blah 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 blah. Doesn't matter. Doesn't matter how to induce this damage to membranes. So it's some oxidation still. In this case by plasma, in other case can be done by some oxygen species. So untreated cells have a very low signal of lipid peroxidation, because anyway there will be some damaged lipids inside your cell, because as I mentioned in some of the previous lecture, there's always some reactive oxygen species inside your cell. It's just a normal part of cell metabolism. So body pie will definitely stain something inside even untreated control cells. But if we apply some cold plasma for 30 seconds, we will observe increase in signal, and on merged image we will see a lot of green staining, which are like connected with oxidation, peroxidation of lipids. So body pie can be successfully used to stain a lot of different lipid-containing organelles in your cells. But so it's a bit just a very small information about what you can stain connected with ROS. And now we can go to colocalization. So colocalization is very important when we are talking about nanoparticles, because if we have some molecular drug, some anti-cancer, anti-tumor drug, there's not very important to find its location inside the cell, because we will definitely know what is the target of this drug. For example, this drug will inhibit some pathways, and there is no purpose to find location of this drug inside the cell, because we know where this drug will go. But in case of nanoparticles, in most cases, this very hard to predict, because they can use some endosomal escape. They can accumulate in some part of cell cytoplasm and so on. So how this colocalization can be executed? One of the simplest example is stain some organelles of the cell with specific dye. For example, here it's rhodamine 123, which is used to stain mitochondria. So this rhodamine dye specifically binds to negatively charged inner membrane of mitochondria. So it's a membrane potential dependent staining. And here you can see some dark field microscopy, and all of these, some of these bright dots, especially the most bright with some yellowish green, yellowish gold color, it's gold nanorods, so gold nanoparticles. And if you merge both of these images, you will observe this. And as you can see, this green, a very pale but still observed green color, it's a mitochondria. And also there's a lot of, well, it's not very good to see, but with your own eyes, well, it's quite better. So staining with staining of mitochondria at the same place as nanoparticles. And this is true because some of gold nanorods can escape endosomes, and because of their surface charge, they can easily bind to mitochondria and damage mitochondria. And because of this colocalization study, we can prove this theory, and we can say that the primary cause of gold nanorods toxicity mechanism is damage to mitochondria. So colocalization is very important when we are talking about nanoparticles. But what other options for colocalization? For example, here, to find location of your nanoparticles inside the cell, not every drug or not every dye is suitable. Because, for example, FDA, very frequently used as I mentioned, but just look on these cells. FDA stain everything, well, basically cytoplasm inside the cell. But since all of the cytoplasm is stained, we cannot understand where this nanoparticle is. So what I mean, if we look on this picture, it's a acridine orange staining, and just look what we see compared to this staining. We see a nucleus, we see some micronucleus inside, we see some ribosomes, because ribosomes are rich with RNA, and RNA stained with orange. So we see a lot of inner structure of our cells. And because of this, not much of cytoplasm is stained, and if you adjust a focus of your microscope, you can easily understand these endosomes with nanoparticles, silver nanoparticles. They inside or outside, or you don't even need to answer this question, because just look on the shape. The shape is very round. It means they are inside, and they're inside some endosomes. And just look on this. It's the same nanoparticles, same culture, just different stainings. And using just FDA, you have no information about endosomes, nanoparticles, uptake, and using just very the same acadine orange. Well, what the similarities? Green color, but not the same result. You observe some endosomes and you observe some accumulation, so on. A bit similar result may be observed with annexin feeds, but still feeds, it's not the best option, because annexin feeds stain only outer on the membrane of dying cells. So just using one dye, you observe a lot of valuable information, obtain a lot of information. But definitely for a proper colocalization of your nanoparticles, you need to use a confocal microscopy, because without confocal microscopy, all of these, like, well, it's around, it means it's endosome. In a good journal, it's not an argument. So for a good journal, you need to perform some confocal microscopy studies. And what is so special about confocal microscopy? It's probably not my duty to tell you how this microscopy works. You just need to understand that compared to simple microscope, optical microscope, confocal microscope have a very good resolution on z-axis. So confocal microscope can split your sample on optical slices, so-called Z-stacks. And these Z-stacks can be, like shown here, separated by 1 micron or even less. And you can slice, for example, if you have some cell, you can slice this cell on optical slices, and after this you can rebuild, like shown on this GIF animation, you can rebuild a whole structure, 3D structure of your cell. And if you have some nanoparticles inside, you can just make a cross-section, middle cross-section, through the, for example, through the center of your cell, and say, well, we have some nanoparticles in the center. It means we have some colocalization with nucleus, some colocalization with mitochondria, and so on. For example, here we observe a lot of nanoparticles in a very big endosomes. It's a gold nanoparticles, or gold nanoparticles inside unicellular algae, for example. So confocal microscope is one of the most powerful instruments to observe colocalization of nanoparticles inside the cell. But let me rest a bit. Let's hope that they will end this bzzz, and so 20 minutes. 20 minutes for testing. Time is yours. I hope everything will work good. If you have any questions, any problems, let me know in chat. Probably I should put a link in chat. If there is no problems, go on. Dear students, don't say any answers yet, and there's 1 minute left. So please finish, because we have still a lot of information to discuss, and I don't want to be that late. Okay, we have like maybe like 20 slides left, so a lot of things needs to discuss. So what's so special when we're talking about some fluorescence and some nanoparticles? Because when we just stain the cells for some organelles without nanoparticles, usually everything goes very smooth, because most of the fluorescent dyes used for ages, not for ages, for years, for decades, and everything quite clear when we're using fluorescent dyes. But when we have some nanoparticles impact, there's a lot of questions can arise. Here we have, for example, some example of two different types of gold nanoparticles. It was 20 minutes of silence during the test. As far as I started the lecture, they started to bzz. Okay. So, two types of nanoparticles. It's gold nanostars. It's gold nanoparticles, well, gold nanospheres with a size of 50 nanometers. And size of nanostars also about 50 nanometers. What's so special about the staining? So here is a DAPI staining for nucleus. It's a propidium iodide staining for dead cells. And this is acridine orange staining, just to stain some DNA also. What we can observe? So on DAPI, we see just a couple of stained cells, nothing special. And well, DAPI, let's say DAPI not as good in staining living cells without fixation. I mean, well, if we increase some gain and exposure on exposition on our exposure, yes, on our microscope, we will see all of the cells, but this is the brightest one. And with propydium iodide, we also see just a couple of dead cells. And every cell is living, and as you can see, there's small bright, small black dots. Yeah, small black dots somewhere, and these black dots is gold nanoparticles. But what we see when we stain our cells after incubation with gold nanostars, almost everything stained with DAPI. We even see some nucleus and some chromosomes and, well, excellent staining. But at the same time, almost all of the cells stained with propydium iodide. And there's so good staining, as you can see. Again, micronucleus and so on, everything is stained. But in Acadian orange, we see some even changes in color. For example, the pattern of staining is completely different and a huge change in color observed. So why this happens? Well, this happens, I just see that only 3 people completed the test, and I know who this people is, and the rest of the people will receive -1 point for every minute they not sending the answers to me. Why this happens? Because you need to understand what you use during the synthesis of your nanoparticles. These nanoparticles obtained by a simple reduction method from a gold chloruric acid with a trisodium citrate, and they are electrostatically stabilized. But these nanostars, because they are anisotropic particles with some like, how it's called, branches, yeah. It's a lot of STAP used, so it's surfactant used to stabilize these particles. And as you remember, I hope, this STAP can damage cell membranes. And what will happen if you have a damaged membrane and you have a dye, fluorescent dye, in your culture media? A lot of dye will pass through this damaged membrane and stain everything. But as you see here, the morphology of these cells looks quite nice. So they are probably all not dead, so they are all alive. And according to MTT assay, they are even more than alive. Because according to MTT assay, as I remember, viability was like maybe 120% or something like this. Why this happens? This happens because the concentration of STAP on the surface of these nanoparticles was so low that it was not enough to kill these cells, but was enough to damage the cell membrane in such a manner that additional amount of fluorescent dye will stain everything, and even damage some nucleus membrane to stain some micronucleus and so on. But why the change in color? Because Acadian orange, as I mentioned, can stain DNA in green and RNA in orange, but at the same time it's pH dependent. And in acidic condition, Acadian orange have more orange color than green color. And since some of the organelles are damaged here, probably some of the lysosomes are also damaged, and the pH inside the cells are more acidic than it should be. Because of this, we have a bit of brownish, like a bit of red added to green. We have some brownish color here, probably. So when you stain your cells, you can easily observe some very unclear, very strange patterns. And to understand what you see, you need to know what your nanoparticles consist of. Sometimes you should understand not only what is the structure of your nanoparticles, but also how works the cells. I mean, how they uptake or excrete your nanoparticles. For example, it's gold nanoclusters. I mentioned a couple of times in previous lectures. So it's just a serum albumin, bovine serum albumin molecules plus 20 gold atoms. So very small structure, like maybe 2-3 nanometers in size. But what's so special about these cells? This is a cell, this is a cell, this is a cell, and this is a cell. And as you can see, there's a lot of the natural color of fluorescence of these clusters is red. But we see a pink fluorescence and we see a lot of blue fluorescence. Why this happens? Well, this happens because of two things. The first one, since these clusters are very small, they can be easily uptaken, and since it's a bovine serum albumin, nanoparticles or cells can easily uptake them and use as a source of nutrients. So cells can eat part of the BCA, dissolve part of this BCA, and because BCA is dissolved in endolysosomes, nanoclusters lose their ability to fluorescent with the red, and they start fluorescing with the native color of BCA. BCA have a number of amino acids, tryptophan, with can fluorescence, so it's some kind of autofluorescence with blue color. So, for fuck's sake. So, what we observe here? We observe non-destroyed nanoclusters with still a red fluorescence. We observed some exosomes or endosomes or endolysosomes with destroyed clusters with blue fluorescence. And all of these small structures, probably like here you see a lot of blue dots. Probably it's exosomes or endolysosomes, something like this. And all of this, if you look precisely, it's not a diffused color. All of these areas of pink, it's like a small circular areas. So all of this is bunch of endosomes. But why pink if natural color is red? Because what will happen if you merge both blue and red? Well, you will observe some pink color. And honestly, there is no pink color. If you look on a spectrum of visible light, it's like blue, it's green, it's yellow, it's red, infrared, ultraviolet. There is no pink in the spectra. There is no such color. But why we see pink? Because pink, it's a merge between two opposite sides of visible spectra, violet and red. And when we have both of them merged in the same sample, we see some pink color. So when you stain something, you should perfectly understand how is this working, how the cells working, and how the light working to interpret your results. I will increase sound a bit, maybe it will be easier to listen to me here. So what can be other problems with image processing? Of course, now there is a lot of machine learning, different approaches to understand what you not even understand, to calculate your cells, a lot of different programs. But before applying all of this algorithm, protocols, programs, and so on, you need to understand by yourself how it's working, because just using some program when you don't understand how it should work, well, can give you some false results. For example, what can be the problems? Here I definitely can see a cell, and I'm sure this is a cell, but it's almost non-visible at this particular exposure. So you should adjust some exposure, but at the same time, on enhanced exposure, this cell will be too bright. So you should somehow adjust exposure of this image in such a way that all of the cells will be visible. In case you have some gel, for example, so your cells growing in some gel matrix, there can be some interference from this gel, for example, dextrane. This dextrane, not a molecules, it's just a border of the gel, and it will be very hard to observe some good fluorescent images when you have some interference from gel structures. There can be some even more complicated things. For example, here we have the HEP2 cells, HEPG2 cells. It's a cancer cells very related to helper cells, and there is no staining. There is no staining, fluorescent staining of these cells. But why these cells are red and the surrounding is green? Because the concentration of silver nanoparticles around these cells was like 200 milligrams per liter, and this is a very high concentration. And because of this very high concentration, probably these nanoparticles can act as some kind of optical filter to filter some, since silver nanoparticles have some optical properties, they can filter the light, and you just illuminate with simple white light and observe some red cells. And this is some artifact of false fluorescence, so you should be aware of this. And sometimes it can be completely ununderstandable results, for example, like this. All of these cells stained just with a single dye, acridine orange. Yes, acridine orange can stain both in green and red, so we see a lot of green, red, greenish, yellowish, brownish, all of the 50 shades of green, 50 shades of orange, but at the same time, some cells have some blue color, and acridine orange can be blue. So what is this blue color here? Probably it's autofluorescence, probably something else. But how I obtain these images? I just left my sample on table for 2 days. So this was unfixed cells, just stained with acridine orange, and I just left on the table and obtained this. But of course, I understand that this is not a proper microscopic technique, but sometimes you can observe something quite ununderstandable, even if you use a proper technique. And the correct answer, what to do with this, how to explain this result, the answer is just remake it, just redo your experiment, because if you cannot explain, no one will explain for you. It's just something wrong happens with your protocol. But let's now proceed to DNA damage. We talked about a lot about DNA damage, and now we need some instruments to assess this damage done using fluorescent dyes. I will already show you some examples of fluorescent staining of DNA damaged or cells with damaged DNA or something. It's some additional examples. So Bordeaux staining, it's brom desoxyuridine and uridine, and this is can be used a natural substitution to one of nucleotide bases, and because of this, this Bordeaux staining, this molecule, can intercalate in the structure of DNA of dividing cells. So if you have dividing cells, they will be stained in green. For example, here we have some control sample and some cells treated with cryozitinib. It's an anti-cancer drug. And as you can see, at a concentration of 10 micromoles, there is no dividing cells. And no dividing cells because, well, they're dead, probably. Also, this Bordeaux staining can be used to stain something on histological slices. Since this drug is for lung cancer, I believe it's some lungs, probably it's alveolar sacs. And as you can see, you can stain with this drug some Bordeaux positive cells. So cells which undergoes division and cells which not undergoes division. This example I already shown you about titanium dioxide and nanocobalt particles, so cobalt nanoparticles. According to different metabolic assays, we will discuss metabolic assays today. So according to metabolic assays, titanium dioxide completely non-toxic, cobalt particles, well, a bit toxic. But if we stain for DCF fluorescence, so DCF it's for reactive oxygen species, if you know, as you know already, cobalt particles can drastically increase amount of reactive oxygen species. So twice here, but not twice decrease here. And What? And also, if we use some other types of staining, it's staining for damage done to DNA. So it's a marker of genotoxicity. As you can see, again, it's increased twice compared to control samples for 15 micrograms per milliliter for cobalt particles, and there's no twice decrease in viability. So DNA is very damaged, but there's no signs of decrease in viability. So it's a genotoxicity effect of these particles. But I already showed you these pictures, but in the same paper, in the same manuscript, they performed other types of tests connected with genotoxicity. One of the most common one is a comet assay. Why it's called comet assay? Well, because it looks like a comet. If you stain DNA in your cells and then put your cells in agarose gel, you can perform some kind of gel electrophoresis, but with the whole cells. And in case you have your DNA not in non-damaged state, in intact state, there will be no tail. In case you have some damage done, for example, by cobalt nanoparticles, you will see a huge tail like a comet, so it's called comet assay. But if you add some antioxidants like catalase or acetylcysteine, you will see a decrease in tail lengths, like here or here. So it's probably a good choice to prove that the main damage associated with cobalt nanoparticles is because of accumulation of reactive oxygen species. And you also can compare it with oxyhydra, dihydra, something. I don't remember the correct name. So as you can see, it's twice increase here, and probably it's twice increase in comet, like maybe it's a cell, yeah, it's twice increase in comet tail lengths. So the length of this comet is a measure of damage done. But they also performed another tests at the same paper, H2X staining, H for, well, it's a family of like a histones. So histones family of proteins which are used as a core proteins to like wrap DNA around them in the in cell nucleus. So damage to this phosphorylation of these histones is a marker of double strain damage. So damage done to both strains of DNA. And using some staining for this, like it's gamma version, phosphorylated version. If you have a lot of phosphorylated histones in your cells, like again cobalt particles, 15 micrograms per milliliter, it's a lot of histone damage, so it's a lot of double strained breaks in your DNA. So honestly, it's a lot of ways to stain damage done to DNA molecules, so it's not very complicated staining. But all of this is direct ways to observe damage done to your cells. So why is this direct ways? Because you directly with your eyes see that this is damage done to histones, so it's damage to DNA. You directly see this is damage done to DNA because of comet tails, and so on. But for example, this was not so direct way, because you see some decrease, but you don't know why this decrease occurs. And only using some direct ways you will see why this decrease occurs. But still we need to discuss this indirect ways, so metabolic assays. MTT assay is the most used in our lab, in other labs, and this is metabolic assay, but an indirect way to observe cell viability, because you need to know how it's working. MTT assay, so MTT, it's this compound connected or have bromide in its composition, and under action of mitochondrial oxidoreductases, this compound MTT can be converted to formazan, and formazan have this violet color, and it can be diluted in DMSO, and after dissolution you can check optical density on spectrophotometer and say something about cell viability. But if you look on this, you should understand it's just activity of mitochondrial enzymes. And these mitochondrial enzymes have almost nothing to do with, well, it's definitely have something to do with cell viability. But why these mitochondrial enzymes have low activity or destroyed? It can happen because of destruction of mitochondria. It can be because of disruptance of mitochondrial potential. It can be some, I don't know, enzyme activity of your nanoparticles. So your nanoparticles, for example, can cut these oxidoreductase enzymes by itself. So it's a lot of ways to reduce mitochondrial activity. Because of this, MTT assay is not very good choice if you want answers. So it's a good choice to very fast screening of activity of cytotoxicity of your nanoparticles. There is also some other examples of metabolic assays. For example, LDH assay. LDH assay works on different principle, but very similar, because again, formazan and so on. If you have some lactate, this lactate can undergoes transformation to pyruvate, and in the process, so it's a part of Krebs cycle, and we will to perform this, we need some lactate dehydrogenase, and this lactate dehydrogenase can generate NADH molecule during this cycle, and this NADH molecule can act as a electron mediator in transformation of formazan. So it's another kind of assessment of cell viability, but the same. Here we assess mitochondrial damage. Here we assess some damage done to Krebs cycle. So probably it's not a lot of information we can obtain from this data. So it's just something happened to LDH. What's next? So it's just LDH. Alama blue or resurgent test, almost the same as MTT, just another compound. So also assessing mitochondrial oxidoreductases. But I will show you. It's basically the last part of the presentation. I will show you why MTT assay not so good, because it can be misleading, and it can lead you to completely opposite direction. Previous lecture I mentioned you a drug called Docetaxel, anti-cancer drug, and this drug can only act, well, at the time I performed this test I didn't use this, so it was just anti-cancer drug. But now I knew that Docetaxel can act only on dividing cells, because it's interfere with tubulin and cells cannot divide. But until division, until mitosis, cell can easily survive even a high concentration of that cell because there is nothing connected with tubulin outside of mitosis. And what we see here, we see a wide range of different concentration of the stock cell from nanograms, nanomoles to micromoles. And we see incubation for 1 day, 2 days, and 3 days. And what we see after 1 day? We see a concentration independent decrease in toxicity. Well, yeah, it very slight concentration dependence, like 70 here and maybe 60 or 50 here, but just look on the range of concentration, it's like 20 times. It's definitely for very toxic, and that stock cell is very toxic, it's not the case. But if we proceed with incubation for 2 days, 48 hours, we will see a drop in viability in any concentration. And this drop is also not so very well concentration dependent. And after 72 hours, we will see another drop. And if we look, it's just almost a line here, almost line here, and almost line here. Well, maybe slight decrease. Why this happens? Because, just because. To prove that these numbers are correct, so these numbers obtained from MTT test. So it's a metabolic activity from MTT test. But simultaneously with this MTT test, it was performed some staining with annexine, and just compare the results. After 1 day of incubation, 24 hours, we see 43 cells are dead according to MTT, but according to staining, only 15% are dead. So 3 times difference. Very strange. And after 3 days of incubation, we see according to MTT almost every cell is dead, but according to annexine staining, just half of them are dead. Why? Well, difference not 3 times, but still 30% difference. How to explain? Because we need some specific number, how much cells are dead. Why this happens? Again, if we look on this, well, first thing is connected with the mode of action. As I mentioned, that stock cell can kill only cells in mitosis. And as we're working with the cells, and these cells was in synchronous state. So it was like a 100% confluence, and all of the cells was in G0 maybe phase. I don't know, but probably they was in G0 phase. So no division occurred. But still, some cells dying, some cells dividing, and after 1 day of incubation, only 20% of cells enter G1 phase, and S phase, G2 phase, and mitosis, and dies, and the rest of the cells, like 60% of the cells, still in G0 phase. So only after 48 hours, some of the cells, like as you see, like a step here, step, cell division, cell division, cell division. So because of the cells, because cells enter in this cell division, like in benches, they are dying like 20. And why there is no dependence? So no dependence from concentration, because this dot cell drug is so toxic that even this smallest concentration is enough to kill everything. So there is no way to add a lot of drug because there is enough of these dot cell molecules to kill all of the cells, even at lowest concentration. So that stock cell need to be to tubulin, and if all of the tubulin is inhibited, no point to add more drug, just no sites for binding, no free tubulin left. So, but other reason, now we're talking about this. Why is the difference between MTT and so MTT indirect ways to observe and annexin direct ways to observe? Why is so huge difference between? Because we need to look on this plot. According to MTT assay, we have a lot of cells, for example, somewhere on this path. So the viability is decreasing, but these cells are not dying. It's just a decrease in viability. Probably some of the cells, 15% of the cells, are already past point of no return. But according to MTT, decrease is like 43. 43 is somewhere here. It's still some time to repair this damage, to accumulate additional ATP molecules. But after some time passed, a lot of cells going through the point of no return, and amount of annexin positive cells also increasing. So between MTT and some staining, there's some hysteresis loop. So the first you observe a decrease in viability, a decrease in metabolic activity, and only after this decrease, only after some time, when the stress is still in place, so nanoparticles are still in your cells, this decrease will result in cell death. Because of this, we have 3 times difference here and only 1.5 times difference here. So you need to know these things. And probably the last piece about simultaneous or combined cytotoxicity. Sometimes it's again from my practice. Sometimes we have a couple of different toxicants in our cell cultures. For example, here we have silver nanoparticles, which are electrostatically stabilized, and they are toxic by itself because of silver. But we add additionally, not in synthesis, just in culture media, we add some step in some, range of concentration. But why we do so? Just to see some combined cytotoxicity. It's just, was just a proof of concept, but in some other cases, you can observe this effect unintentionally. For example, if you have some, again, toxic nanoparticles covered with some surfactants because of the synthesis method used. So what was the result? The result was that after 1 day of incubation, at the lowest concentration of both STAP and silver nanoparticles, we observed increase in metabolic activity by 30%. After 2 days, we observed increase even by 40%, and in the larger concentration. But after 3 days, almost everything dies. Why this happens? Well, the answer quite mysterious, but I can only suggest that all of this phenomena happens because of depletion of glutathione inside the cells. Because it's again, look at this plot. If we have some accumulation of, in this case, Rexy species, but it can be any other types of toxicants. And to, let's say, to eliminate this threat, the cell will release additional molecules. So release some, put some additional power to eliminate this threat. And but the capabilities of cell are limited, and because of this, after we still increasing this amount of toxicants, like for example 1 day, 2 days, and finally 3 days, there is no more reserves left in cell and cells dying. So probably that's the explanation. But still, to understand this, you need to know how the antioxidant system works, how the cells works, how they behave, and so on. And the last slide today, just again a bit of examples to show what can be the problems when you are assessing viability of your cells using some metabolic assays. Examples with again silver nanoparticles. For example, look on this culture well plate with silver nanoparticles. It's a control sample, and look at this. It's 200 milligrams per liter of argentum particles, and it will be very, very hard to read optical density after MTT assay because of a lot of nanoparticles accumulated, adsorbed on the surface of culture plate and on the surface of your cells. And there is no way to wash these nanoparticles because they can be on the surface, on the membrane, in the membrane, inside cells, and they will interfere. And this interference, in most of the cases, cannot be calculated because nanoparticles, especially one with optical responses, they can undergo different optical changes. So they can change color, they can change, as I showed some slides before, somewhere here, for example. So they can change color of white light, they can change color of MTT, and it's very, very hard to predict. Yeah, okay. So it's very hard to predict what happens, what can happen to your sample, not your sample, but with your assay. Well, it was the last slide. So what I want to say, what is the conclusion about all of this? The conclusion is that when you are assessing toxicity of nanoparticles and you don't know what is the mode of action, what is the cause of cytotoxicity of your nanoparticles, you need to perform a multiple test. You need at least understand that you need to stain your cells with some fluorescent dyes to check a membrane integrity. You need to perform MTT assay as a base. You need to observe your cells with just optical microscope. And you should be very careful when you're talking about the real cause of cell death. So you shouldn't say because of DNA damage, because I don't know, because it's silver nanoparticles, some reactive oxygen species will damage DNA. No, you cannot say that because before you stain with specific dye. So it's a lot of things can be also discussed additionally, but I hope some of this basic information was useful for you. And if you have some questions, you're welcome. And after the lecture, please enter a Zoom session. So I will listen your presentation, and please today no longer than 10 minutes per presentation. 10 minutes limit. So if there's no questions, thank you for attendance. Next Monday will be the last lecture, and I believe Wednesday next will be exam in Zoom. So I will give you all of information you need. After the last lecture. Well, after the last lecture, there will be some kind of consultation. So if there's no questions for today, again, thank you. We'll see you in Zoom meeting.